Hello students, welcome to the lecture 3 of week 2. So in this lecture we will cover the different sequence formats that we use to store sequence, share sequences, right. So these are the standard formats that you should know because you will be downloading data in these formats such as FASTA, GenBank or EMBL or the next generation sequencing reads format, or you will be producing data or sharing data with other users in this format. And then we will also see the different genomic databases that you can use to download this data, right, and also how to submit this data to those databases. Okay, so why do we need a sequence format? Because you have to keep the file format uniform and unique so that it can be easily identified by any other person, right, and the tags are defined. You know that what is the sequence, what is the labels associated with that sequence, right, what are the annotations of that sequence. For example, here if you see, like there are two common file format which are shown. For example, the FASTA format. Okay, so for example, here you can see this identifier which is a greater than symbol. So with this identifier, after this you can write anything in the annotation. Okay, so annotation can be a user defined thing or sometimes fixed as per the database or the sequence format, or okay, so in this line you can put anything that is associated with the data, or you can say metadata of the data, right, and in the next lines you can put the sequences, right, okay, like this. Okay, and similarly in a multi-FASTA format we will put multiple sequences one after another. So like this is one block, okay, and the another block starts with this greater than symbol, then annotation and the sequence again. Okay, so this is a typical like a FASTA format which is most commonly used because it is compact, gives the minimal and useful information about what is the sequence and what are the identifiers, right. And the other one, on the other hand, if you see the format which is a GenBank format, here you are defining a little more. For example, you will tell that okay, like different tags are here, locus, definition, origin. We will see some more later, right. So here I am writing that okay, this is sequence 1 containing 16 base pair, and basically there is a also checksum index which is validating the sequence. Okay, the basically the sequence here starts with origin word and ends with this backslash, 2 backslash, and in between the sequence. And this sequence is also generally written in a block of 10, so 10 characters each, okay, 1 to 60 for example, right. Whereas if I write the sequence in this FASTA format, it may be a continuous sequence or it could be a block of 60, and then the next line starts and then again you write the 60 characters. Okay, so basically one format which is FASTA format is to keep the sequence compact, right, and the basically the annotation compact and the sequence beneath it. Okay, and it can be readily used by various scripts and softwares. Another one is the detailed format which is the GenBank format. So for example, here we are defining a little further. So this is sequence 1. This is a protein sequence, right, amino acid sequence basically, and you can see that there is no end defined, but basically wherever the next sequence starts, greater than symbol and sequence 2, the first sequence ends. Okay, so we don't need to define an end here, and so you can see that it is right now I think 60 characters per line, but you can also put it in a single line and that is also acceptable. Okay, or you basically for clarity or for like more better readability, you can split that into new line characters after 60 characters, right, and basically when you write multiple of these together, it is called multi-FASTA format. Okay, so this is the most popular sequence format. Now the GenBank format in detail, if you see that basically this GenBank file is downloaded from NCBI. Okay, and so basically I search this ID or this basically some annotation of the line that what you want to search in the NCBI, and then you got the option to download the sequence as a FASTA format or in the GenBank format. So you can see here that in the Genomic format I can get all the information that may be required for my further research. For example, what is the locus, the definition, that is the title, okay, the accession number, then the version. You can see that AB 132.037.1. So this means that this is the version number 1. So if there is some modification later, it could be 0.2 or 0.3 or 0.4, okay. Then basically the keywords, like if the user can define multiple keywords, okay, it's a satellite sequence for example, right. And then Homo sapiens, the like what organism it is coming from, okay, and the detailed taxonomy of that organism, what authors contributed this sequence, okay, the title of the study that reported this sequence, where it is published, okay, what is the PubMed ID, okay, and then again the authors in that sequence, because it could be in that publication. So it could be that the sequence submitters could be a group which is more experimental, or okay, and the publishing group may be little different, okay. Sometimes you outsource, sometimes there is a team that generates the data, but may not be a part of the actual publication where more work and analysis is required, okay. So and then basically the journal and okay, so you can see that here we are defining that features. This is important, that in the features we say that okay, what is the source and taxon and what chromosome it is coming from, okay, location exact on the chromosome, right. And then basically what primers were used to amplify it, it's a repeat region, right. And like this is a sequence, so where it is 60 character block, each block is 10 base pair, 1 2 3 4 5 6 and blocks are there, okay, 60 characters per line, and then you go further to 1 to 333 base pair, okay. So this is a more detailed format which gives you plenty of information about from where the sequence came from, who generated it, and whether it has gone some modifications, it is published or not, right. And actually you can put much more information also, because there are several more identifiers that you can use in GenBank, okay. Now for the same sequence, if I download from NCBI, so this was the sequence that we were dealing in the previous one, okay, the same sequence. So if I use that as a FASTA format now, so it is just the accession number, the annotation line and the sequence in the FASTA format, right. So basically the annotation line and the sequence, okay. So this is much more compact. So when you want to process files for analysis, you will prefer FASTA format, but you want to be more gain more knowledge from that sequence, you will go for the GenBank format, okay. So basically in NCBI, if you go, you will have the option to then basically you can on the right side you can see the send to option, where you sending as a file in the format you are defining. Here I can define GenBank, FASTA or other formats also, and then create file. So this file will be stored in your system, okay. So generally when the file is stored in your system, when you double click on that, you may not be able to open the file, okay, because the files are not in text, okay, txt, they do not have that extension which the normal Windows machines can read. So what you can do is, when you download the file, after that you can put .txt extension after the file, then you will be able to open that file, okay. Then another format is Swiss-Prot format. Swiss-Prot is a very important database that houses the curated protein information, and you will definitely be using it. So again, in the Swiss-Prot database, you can get the sequence in FASTA format, like the way we have seen in the previously, or the Swiss-Prot also have their own format, okay, which is also descriptive, like for example ID, accession number. So earlier we were using the entire word locus, feature, and okay, broader word, but here we are just using like 2-letter word of ID, AC, date, DT, description, DE. So basically here I am writing that what is supposed to come over there, okay, like accession means like accession number, okay, but in the actual file you will see that this is more filled up with the actual information. So date will be mentioned over here, description, gene name, organism, and like that, and basically the sequence header, okay, then the sequence and the termination line. So which is more similar to GenBank in this part, where it was origin in GenBank, origin in place of SQ, and // which is always also present in the GenBank format, okay. So basically if you see here, each line begins with a 2-character line code, okay, which indicates the type of data that is contained in that line. And then if for example some data is not available, for example reference number or reference position is not available, you may like to skip this identifier RN or RP in your format if you want to create it, or you can put some NA or not available in that position. So both are acceptable. Okay, then these days we are producing a lot of next generation sequencing data, NGS, next generation sequencing data, right? And there are two common NextSeq platforms, which are the Ion Torrent and the Illumina. Okay, so both produces data in their own format, and basically when they are producing the data, they want to include some information about the sequence quality, right, along with the file. So for example, this is a QSeq format, okay, which is typical of the Ion Torrent system, right? And okay, so it is starting with some file name underscore qseq.txt, for example. So this will tell that this is a QSeq format. Okay, then it is a plain text file for sequence read, and here if you see that it is including the information about the machine number, run number, lane number, okay, machine, that which machine was used to generate it. Okay, when you sequence something, it is called runs, run 1, run 2, run 3, run 4. Okay, so sequencing run. So one, basically when you set up a sequencing you can say cycle, so it is called a run. Okay, so basically whenever you are sequencing, each time you are sequencing, it is called one run. Okay, so run 1, run 2, run 3. So it tells you, so machine is recording that how many runs have happened so far in that machine. So run number, lane number, like in the flow cell where exactly your basically sequence was located, which corner of the flow cell. So it's a glass slide kind of thing, right? The tile number and coordinate in that, so that you can easily locate that which part of that flow cell your sequence was located. For example, okay, so it was located in left part, so it will tell you that the coordinates of the left part. Okay, so this happens because like in the end we are getting a sequence. So if in that corner or that part you are not getting good quality sequence, then you can always go back and check that whether the camera did not work in that place, or there was some other liquid, basically the fluidics issue or something like that happening in that area. Okay, and then the quality sequence basis, quality, the read number, sequence basis, quality and filter. Okay, so read number, the sequence basis, you can see that it is .ag. So basically in this space no sequence was reported. Then it was a .g . That means nothing was available over here, and the quality is written here like, okay, B. Okay, so qualities are generally like this B will actually refer to some quality score or value. Okay, so there is a table that is associated with the formats, and yeah, you can also go back and check that, okay, B refers to what quality value, C refers to what quality value, okay, like that. And in the end you have 0, that means whether the read passed the filter. So basically no, so that means 0 was mentioned here. So that means it is not a good quality sequence and it has not passed the sequencing filter. Okay, so right. So basically similarly I'm just explaining that further machine name, run number, okay, unique identifier of the sequencer and the run on the sequencer, okay, then lane number, tile number, coordinates and the sequence quality, right, which we have mentioned. And there has to be one record per line, each records represent one read, okay, and right. So basically this is the QSeq format, which is one of the common formats, but mainly like what you get mostly in the data set is the FastQ format, which is this Illumina FastQ format. So okay, so similar to QSeq, Illumina FastQ also reports the sequence, some information about the sequencer and the run number and other things, and also the actual sequence. So basically you can say that it's a FASTQ format uses 4 lines per sequence, okay, 1, 2, 3 and 4, right. 4 lines are there per sequence, right, and basically line 1 begins with a @ character. So in the FASTA format you saw that it was the greater than symbol, that was the starter, so that you will know that it's a FASTA format. And for this FASTQ format, the initial character is @, okay. Then there is a sequence, okay, right. It could be like for example the sequencer has produced 150 base pair read, so it will be 150 base pair in, okay, length. If it is 75 base pair, it will be 75, 50, 25 and other options which are available with the sequencer, okay. And after this there is a standard like you can say delimiter, or between the two the sequence and the quality values, okay, blank line containing plus in between, okay, and then the actual quality values. So for example, the G quality values is exclamation, A is quotes, T is star, okay, T and this, okay. So each of this character actually refers to a quality score, right, again which you can go and check, or basically it is inbuilt in the software that you are using for quality filtering, okay, and it will read or extract the value from there and determine that what quality your sequence was actually, okay. So basically you have seen that in 4 lines you have told that, okay, what is the sequence ID, which will give information about the machine, run number and other things, the actual sequence, a blank line with plus at the start, okay, which is standard, okay, it will not be minus or something, it is always be plus, right, and the quality values for each character that you have reported in the second line, right. So this is the typical NGS FASTQ format, okay, which you can get like in .fq, .fastq, these kind of extensions could be there in those files, right. So 4 lines, @ symbol, the actual sequence, a delimiter and basically the quality values. Okay, so then there is a general feature format, like sometimes you want to prepare information for a software, or the software tells you that, okay, you can put the data in this format, or right, for example simply like gene name, start, end and this strand. So there are like 4 columns in this: gene name, start, end, strand, okay, like this is sequence 1, EMBL, start codon, start, end, and basically okay, some other information, that plus strand and like 0 may refer to like some value or something like that, okay, the attributes or comments, right. So it is not fixed that you will be putting information in this way, so you can define your own format, that okay, you have to give me information in like 4 columns, which will mean first column will tell this, second this, fourth, second, third this and fourth this, okay. So you can put your own columns or information according to that. You have to fill up that table. So for a program you can keep it constant, and similarly the program that you are using may also ask you to put the information in that format, okay. So this is a general feature format that sometimes you have to prepare, or sometimes the program will ask you to prepare this. Then there is another format which is more descriptive, that is the XML format. So it is basically full of tags. So like for example, just one sequence like this one, if I export to XML, so this I can export to GenBank that we have seen earlier. I can also export to FASTA, which is like 2-line format, greater than symbol, annotation and the line, and the XML format, okay. So the previous 2 files had the issue of the extension and what software will open it and like that, but this is a portable format which is called XML format, okay. So extensible markup language, okay, which is a more descriptive and platform independent format, you can say, okay. So it is more both human readable and machine readable format. So it will be like you are arranging things in the form of okay, these are tags like bioseq, and you can define the tags whichever is, sometimes information may be there, sometimes information may not be there for that tag, right. So basically it will give complete information about whatever is information available for that in the database. Okay, so it's a bulky format, but it is very extensive, and then you can, for example, you are interested in this part, that which organism lineage it is coming from. So you can know that okay, there is a tag called organism name underscore lineage. So I will write a program that can extract this tag particularly, okay, in that file, and give me the information about the lineage, right. So that way it is very helpful for computational analysis. But the issue you may face is that the FASTA format may be like a few KB, but this one could be very bulky, much bulkier, because of this, the way they have arranged the information. Okay, then there is we have said read that proteins are the structure is solved, and basically these structures are available, or the information is available in protein data bank. Okay, so PDB format, so which is more about the what was the protein, how the structure was solved, okay, who did it, like the header, the title, that this is the extracrystallographic determination of a collagen like, okay. Titles are in the 2 lines. So basically if it is 3 or 4 line also, you will write title like that, 2 peptide with the repeating sequence, right. And that how the data was generated, experimental data, the authors who generated this data, okay, and some remarks about the data, and then the actual coordinates of the that you got from the X-ray crystallography, and right, so that it can be used by softwares for basically constructing or this structure again, because it has all the coordinates for each and every atom, right. And also by for molecular dynamic simulations. So this is a important format for when you are doing the protein structure work, okay, and this is available in protein data bank database, okay. So we have seen the sequence formats, and basically now we know that what are the commonly used formats that we will encounter, or we have to create, or we will retrieve from various databases. Now what databases will contain those, and how will you submit your data over there in these formats, or how will you retrieve your data, okay, this is covered now in this section. So like this is like, as I mentioned earlier, that NCBI is one of the comprehensive resource of the data. You can get all kinds of data, whether it is sequence, protein, DNA, RNA, protein, markers, okay, expression, okay, linkage, inheritance, all kinds of data people submit over there, but you have to download that. So like there are, like for example, for genomic data, NCBI data sets are available, like you can enter the species name here, okay. Some examples are, for example, here you can type salmonella or something, or human in this portion, or you can use either the common name or the scientific name. Better to use the scientific name so that you get the specific information, okay. Like this is common name is Chinese hamster, but scientific name is little extensive. So okay, so you can search here, and also okay, so you can also search the genomes that you want to download. So some commonly used genomes are available, okay, in this search in this suggestive options. Otherwise you can write the complete name or taxonomic name for that, and you can get the information, okay. So when you are reaching to this page, you will be getting information about the various chromosomes it may contain, okay, the nucleotide files, the protein files, okay, or any other information that is related to that, okay, genome. Now when you are downloading data, you are also you should be particular about what version of the data you are downloading, okay. What is the release date? For example, it is October 16, 2023. The release is this, right, and this is the assembly 2.0, okay. So basically this will tell that okay, for this particular genome I have downloaded this assembly, the release number is this, and the release date is say October 16, 2023, right? And you can see that there are some which are like little older, also October 2, which is constantly updated also. So for one particular genome, you may get multiple releases and multiple assemblies. Okay, so you may like to stick to a particular assembly for your analysis, right? Because if you are a PhD student or doing some research, you would like to spend like few years on that data. So you don't want to go to another version of the data, because everything, all coordinates, information may change in the other version. Okay, you can use the other version for further validation or suggestive or like that, but still you would like to stick to a particular version that you are using for your analysis. So that is why this assembly, release and release date are very important, which you should always keep written somewhere, so that you know that what version of the data you were using. Okay, then when you are saying that I am downloading data, basically somebody might be uploading the data also, and basically these are the researchers across the world who are doing sequencing projects, or it could be DNA RNA sequencing, or protein data, right, or anything that is coming out of their experiments, which they should submit to NCBI, for which the option is available at NCBI. Okay, so for that you have to submit the data on the NCBI SRA. For example, the read data you will submit in NCBI SRA. Okay, so it is important to submit, because for any data that is produced by a person, it has to be submitted to NCBI before the publication, because it is also expected by the reviewers that the data is available publicly, so that otherwise people will be holding data to their groups and it will not be available for further research. Okay, and generally you do this after the publication, so that at least you have authenticated that okay, this data was publishable and the results were obtained were reliable, peer reviewed. Sometimes you may also like to open data if you are running a very large project, publicly funded project, so that people can regularly access the data. While you are doing research, they can also do the parallel research. Okay, so data upload can be done only after you have created an account, because otherwise anybody and everybody will be uploading something. So there are strict checks that what you are uploading, and it is not that any junk data will pass through. So you have to be very particular, and your account may be suspended for any such negative activity. So the data that you have submitted will be verified by NCBI and will be given an accession number. Accession number is a unique identifier for a data. Okay, or it could be a sequence, it could be the complete project. Okay, that may contain several sequences, right? And this accession number may also change later once more information is available for that. So accession numbers do retire or change or get upgraded, but there is always a way to go back to like a new accession number for a older accession number, and similarly to the old accession number, because that information is available at NCBI. Okay, and then you can select the option that you want to release the data immediately that you have submitted, or at a later date. Okay, so that I think up to 4 or 5 years you can hold the data, right, for release, because that is the time that you may need for your analysis to complete. Or you can or as soon as the publication is done, NCBI will keep track of that, and basically it will release the data, because in the publication also you have mentioned that accession number. So whenever that accession number goes online, NCBI will detect that, and they will release that particular data after duly informing you. Okay, so so basically at a later point of time or just after the publication, it can be opened. Okay, and or can also be released immediately, depends upon the data and the purpose. Okay, and basically this sharing and accessing of data by researchers okay, is the way that we do science, and a community based approach where somebody is generating, somebody is accessing, and then what they generate, you will also take benefit from that data or the results. Okay, so this is the NCBI homepage. So here there is an option to submit the data, okay, and also the option to download the data. So right now we are dealing with the submission part. So by using this option, you can submit your data. Okay, just click on that. So basically go to the NCBI homepage, submission portal, okay, go to the SRA section, and then you have to basically sign in, right? So you can make your own login for NCBI, or you can also use some ORCID ID or other IDs which are allowed by NCBI for signing into the NCBI database, right? So okay, so you will sign in for uploading the data, right? So this is the SRA database. You will log in, okay, here, and then uploading and start the upload, okay. So submission portal link, you can also go directly, right? This is submission portal link. So what do you want to submit? So basically it will asking you that it's a DNA, RNA sequence, or some expression data, or anything that you want to type, okay, right? A genome, 16S ribosomal RNA, SARS-CoV data was very important recently, so that is also an option, okay. So then okay, after that you will say that okay, I want to submit, right? So this is the second page that comes, and SRA is the world's largest resource of the data that is submitted by the users, okay. So it accepts data in a particular format, and which is the file format guide is there, which you should read before submission of the data, okay. The files can be in a zipped format, okay, gzip or bzip2. So these are the compressed file formats, like you have definitely done a zip of your file in Windows. Similarly in the Linux we do gzip. I think in Windows also gzip is allowed now, okay, or bzip2, which is even more compressed format, right? So but zip is not allowed, okay. So then file names has to be unique and does not contain any sensitive information. So you have to particular about what you are submitting, and basically you should not refrain from submitting any junk thing, because it will be unnecessary burden on those who are verifying, and also to the community, right? So basically what you are submitting, some prior information has to be submitted even before that we will see later, okay. The file sizes, if cannot be more than so basically there is a preload option if you are uploading very large data, more than 10 GB or more than 300 files, okay, and each data has to be in a single folder. They can also give you an access to their FTP folder, and you can submit data directly there. And the folder that you have created, you must complete your submission within 30 days. You may also request for extension, but otherwise if you do not say that okay, the data upload is complete, they will remove your folder after 30 days, right, if the data completion is not completed from your side. So this is to ensure that there are no partial or incomplete folders, and they also have a cycle to basically validate and verify the data, okay. So you sign in using either ORCID ID, which is these days, like for researcher, ORCID ID is important, which tells you kind of brief profile of what you are, where you are, okay, your publications. And this ID is also acceptable for accessing the various journal pages and submitting manuscripts, and also to sign into NCBI, okay. Or you can also access through institution, Google or Facebook, and which are the acceptable options here, okay. Or you can also create your own NCBI ID also, right? So then after reaching to sequence archive page, you will say I will want to submit go for a new submission, because when you start something you always have to submit a new submission, because if you are already a part of a consortium, they have given you sub access in terms of login password, and they've also defined the bio project or the bio sample, which are supposed to be for in which you have to upload the data, then it is another case. Otherwise you will also do a new submission, okay. So where you will define that what data you are submitting, okay. And sometimes you also want to check that whether something like that is already there, so you can do some query and check that something like that exists or not, okay. Then you have to provide information about submitter, the name, common like email address, name, and alternate email ID, okay, and what institute you belong to, your complete address, okay, standard things, right. and whether you have created a bio project or not. If you have already created, it's okay, otherwise you have to create a bio project, right? Right, for many cases. And then if it is already there, you can type it here and it will, you know, fetch that record. Okay, then even same for bio sample thing, right? And then the option that when do you want to release the data, okay, which I mentioned, you can release immediately following processing, or on a specified date, or after publication, whichever is first. So if it is published before the date you have mentioned for release, it will be open to the public soon after the publication. Okay, then the project title, what is there in that? It could be, project title could be a brief line, it could be like your publication title, or something to define that what is contained in that. For example, like turmeric genome, okay, could be one title, or turmeric genome sequencing by Illumina platform could be one title, like that. Public description, that what, how public should know that, you know, or access this record. So you can tell that you know, the first genome publication of genome sequencing of turmeric, chromosome level information is available, high C was used, and like that, whatever you want to put in that description. Okay, relevance will get you some you know, options, and you can see that what is fits best to your project. Okay, if it is a part of a larger initiative, or just you are doing on your own, so you can, you know, as per the project you can define. Okay, and some description or URLs if you want to put for external links. It could be a link to your page or some agency that for which you are doing the sequencing, and like that, right? Like consortium name and these grants information, right? And then you will you know, start uploading it. So okay, so basically here you will define that what organism data you are submitting, and you know, some more details about from how you extracted that, okay, where do you find that, the coordinates of the exact location. Okay, and basically this part defines that what kind of data you are submitting. It could be eukaryotic, prokaryotic, okay, a genome, or a marker sequence, or you know, so virus data, bacteria data, like that. Okay, then on the next page you have to give details about the what you are submitting. Okay, so they already have some predefined Excel sheet, like sample name, sample title, by project accession, organism, isolate number, cultivar, ecotype, age, developmental stage, geographical location from which you got the sample, tissue, etc., etc. So basically many times you may need this, you may have this information, sometimes you don't have, so you can leave some things blank, or you can put none over there, but you should try to fill as much as possible, because this information will be helpful for others to know that how the data was produced, from where it was produced, and okay, like organism, cultivar, and those things, right? So okay, so here there are options that you can use a built-in editor, like which is already defined, and you have to just fill up those you know, Excel sheet. Okay, right? Or you can upload your own Excel sheet where you define your own headers based on you can check the NCBI file format and what headers are allowed. But I think this is a convenient option, and this is generally, you know, we have also used and preferred. And you can, here it is also mentioning delete submission, that if you do not want to submit or delete it, right, then you can also delete using you know, this tab. Okay, so okay, so this basically, this is the table editor. So then after that you will be uploading the files, right? And after successful uploading, they will review and you know, provide an accession number. Generally within 24 to 48 hours your accession number comes, if it is not a weekend or a holiday, right? So this is all about the data submission. Now how to access data. Okay, so basically there is NCBI FTP site from which you can download the data, or using some application like FileZilla. Okay, so you can download the data using the NCBI site FTP, right? So what is FTP? Basically it's a file transfer protocol in which you have a login name and password. Okay, so login name is basically okay, the username or the name that you have created for your account, @ symbol and the IP address of the host. So okay, so like ncbi.nlm.nih.gov, okay, could be the host name, and or you can check at the NCBI site, or it could be within internal transfer you will be defining the actual IP address. This is just a hypothetical IP address, right? So FTP name at the IP address, and then it will be prompted with a password thing, and then you write the password. Okay, so this way you are connected to the machine, and then you can basically put your data by saying put this file which is there in your local directory. So basically the external machine is now connecting to a local folder from where you actually initiated this FTP thing. And how do you get to the FTP thing? Basically, in the terminal of Linux or command box, you can connect to external machine and say FTP this, right? Name at the IP address. Okay, so basically it is just for putting something or retrieving something. Okay, and also sometimes we will discuss FTP in detail when we do the in the next class, when we do the Linux commands, right? So basically NCBI also provides an FTP access by which you can put the data or retrieve the data from there. So this way you don't have to use the HTML page and do this manually for multiple files, and you can download the entire folders or multiple files using FTP. Okay, or but okay, so FTP you are doing, so you have an internet access and connectivity, but if that connectivity breaks in between, your FTP will be broken, the link will be broken, and then you have to resume or check that okay how many files you have downloaded, and again go back from start and like that. So okay, in that case basically what you can do is you can do FileZilla. There is an application, you can download this client, and then here you can tell that what host like NCBI NLM.gi or basically the machine ID, okay, the IP address, username and password, and the port 21 or 22. So 21 is for FTP, 22 for secure FTP, which is more preferred these days, right? And then connect. So basically you can see this is the folder from which you are logged in, your local folder, and this is the folder, remote folder, which you have connected to. So you can basically just drag drop this thing here on this thing here, and you can do the file transfer, and you can also view the progress in the progress bar below, that what has been done successfully. Even if the internet connection breaks in between, it will try to reconnect and resume from the place where it was broken earlier. So this way it is more safer and easier to download. So FileZilla is very good application for this. Okay. Okay, so what we learned in today's lecture is like what are the different sequence formats. The commonly used ones are FASTA format, Genomic format, EMBL, FASTQ, right? And the different the genomic database, particularly the NCBI, and what do you mean by the sequence archives, and the release date, and other summaries of the sequence. NCBI SRA, sequence read archive, and how to submit the data, how to retrieve the data from there, and two common applications for this, which are FTP and FileZilla, right? So this is all for the today's class. Okay, thank you.